Have you ever thought about what happens if your cells just, forget what they're supposed to be doing?
Like a sort of cellular amnesia.
Exactly. Imagine if overnight your highly specialized liver cells or your protective skin cells suddenly lost their job descriptions and they just started acting like generic temporary workers.
Right, which completely reframes the whole conversation around getting older.
It really does.
Yes.
What if getting older isn't about the machine parts of your body wearing out, but, well, the software getting corrupted and losing its specific instructions?
It's a profound shift in how we view the biology of aging. We are so conditioned to that wear and tear model, the idea that joints rust and filters clog.
Yeah, the mechanical failure idea.
Exactly. But the concept that our cells are experiencing this fundamental amnesia, it changes everything about how we look at chronic disease.
And that software corruption is exactly our mission for this deep dive today. We're exploring this massive concept called epigenetic information loss.
It's a fascinating area of research.
It really is. And we're pulling from two incredible sources for you today. The first is this sweeping background paper that analyzed 30 different human tissues to show how this cellular amnesia is, well, a common feature of aging and multiple diseases.
And the second source is incredibly specific.
Right, a manuscript that zeroes in entirely on how this happens in the liver. And spoiler alert, it reveals how metabolic interventions like fasting might actually hit the undo button on this whole process.
Which is the really exciting part.
Okay, let's unpack this, because the idea that aging and disease might not be a strict one-way street, that changes everything.
It really does. Our goal today is to go beyond the surface level changes. We want to dig into the mechanics of what happens to our DNA as we age, why it matters,
And how our cells might actually retain a hidden memory of their youth. Right, just waiting for the exact right environmental signal to wake it up. So, I want to start with that hidden memory, because before we can understand how to reverse aging in the liver, we really have to understand how a cell knows what it's supposed to be in the first place.
That's the foundational piece, yeah.
Like, every single cell in your body has the exact same DNA blueprint. Your brain cells have the same basic genetic code as your toenail cells.
Right. The genome is identical across almost all your cells. It's the master blueprint.
So how do they end up so utterly different?
Well, the way those cells differentiate themselves is through the epigenome. Remember, epi means above the genome.
Right.
And the primary mechanism we are looking at today is DNA methylation. You can think of methylation like a complex system of redaction markers and sticky notes on that master blueprint.
Telling the cell which parts to read and which parts to just ignore.
Exactly.
And structurally, just so we can visualize it, these redaction markers are physically attaching to the DNA ladder, right?
They are. Think of the DNA ladder, and along that ladder are specific rungs called CPG sites.
Okay, CPG sites.
Yeah, these are just specific chemical locations on the DNA strand where these methyl groups, our redaction markers, can securely attach.
Yeah.
So the background paper we're looking at analyzed 30 different human tissues and mapped out what they call tissue-unique methylation sites.
And I found a ton of them, right?
Nearly 88,000 of these specific spots across the genome.
Okay, wow. So these 88,000 sites are basically the defining features of a cell's identity.
Exactly. At these sites, a particular tissue will either be methylated much, much higher than the rest of the body.
Which they call unique high.
Right. Or much, much lower than the rest of the body, which is unique low.
Okay, so that sharp contrast is the epigenetic information.
It is. It's what makes a kidney cell fundamentally operate as a kidney cell.
So if I'm tracking this, a unique low site means the redaction marker is removed, the DNA run is open, and the cell is actively using that specific instruction to do its specialized job.
You've got it.
And a unique high site means that specific instruction is heavily redacted, locked away, because, well, that tissue absolutely shouldn't be using it.
Exactly. That sharp contrast high where others are low, or low where others are high, that is the defining signature.
But something goes wrong over time.
Yeah, during disease and aging, these incredibly sharp, unique patterns just start to degrade. The researchers found this phenomenon they describe as regression to the mean.
Regression to the mean, meaning everything just sort of becomes average.
Exactly. The unique high sites lose their redaction markers and drop down toward the pan-tissue average.
Oh, wow.
And meanwhile, the unique low sites inexplicably gain redaction markers, rising up toward that same average. The distinct peaks and valleys of the cell's identity just flatten out.
So it's kind of like a highly specialized Michelin star sushi chef slowly forgetting their exact recipes, losing their delicate knife skills,
Hmm.
and just making generic fast food burgers like everyone else in town.
That is a perfect way to look at it.
I mean, they still have a kitchen, they still have ingredients, but the unique identity of what they do is just entirely blurred,
And when that Michelin star chef becomes a line cook, the tissue loses its specialized edge.
which has to cause problems.
Big problems. The researchers found this happening in multiple scenarios. Like in chronic kidney disease, the unique methylation signature of the kidney steadily erodes as the disease progresses.
And they saw it in skin too, right?
Yes, they looked at human skin, specifically comparing sun-damaged skin on the forearm to sun-protected skin from the underarm.
It's interesting they used skin because you can literally see the difference between sun-damaged and protected skin.
Right.
You're saying that physical wrinkling or weathering correlates with this deep software blur.
Absolutely. In the epidermis, the outer layer of the skin, chronic sun exposure, caused those unique skin methylation sites to regress to the mean.
That's wild.
The environmental stress literally caused the skin cells to lose their epigenetic information.
But wait, does every single disease cause this blurring? Because if a cell is under stress, it stands to reason it might just glitch out. Does every sick cell just forget what it is?
That's a great question, and the researchers tested that assumption. The results show it is not a universal rule.
Really?
Yeah. They used autism as a negative control, analyzing brain tissue from autistic individuals. In that case, the brain cells did not show this regression to the mean.
Oh, fascinating. So the neural identity remained distinct.
Distinct and sharp. So it's not just a blanket reaction to any biological atypicality.
Okay, so there's nuance here.
And it gets even more complex. When they looked at the pancreas in patients with type 2 diabetes, the unique methylation sites didn't regress to the average at all.
Wait, what did they do?
They actually diverged further away from the mean.
They diverge. So instead of the pancreas forgetting it's a pancreas, it like doubled down. Is it panicking and trying to overcompensate?
That is the leading theory, yeah, a compensatory hyperfunction.
Oh,
The pancreas is under immense stress to produce insulin in a diabetic state, so it may be aggressively altering its epigenetic landscape, hyper-specializing to fight the disease before eventually failing.
that is incredible. But the key takeaway from the background paper is that regression to the mean, this specific smoothing of the landscape, is strongly and specifically tied to age-associated decline and chronic degenerative diseases.
Precisely.
Okay, so we have this established mechanism. As we age, or as certain tissues endure chronic damage, our cell's software gets blurry. They regress to the mean.
Right.
Let's pivot to the liver manuscript. Think about your own liver right now. It's sitting there filtering your blood, managing your metabolism, and it's famous for its superpower to regenerate.
It is the ultimate regenerator.
Right. You can lose a significant portion of your liver, and it will literally rebuild itself.
Yeah.
So does an organ with that kind of regenerative power still fall victim to this amnesia?
You would think its regenerative capacity might protect its identity.
Yeah, that makes sense.
But the liver has thousands of these unique methylation sites. And when researchers analyzed multiple liver diseases like cirrhosis, non-alcoholic fatty liver disease, NASH, they found the liver does not protect its software at all.
Not at all.
Astonishingly, 99.6% of liver-unique methylation sites regress to the mean as liver disease progress.
99.6%. That is virtually all of them.
Almost completely wiped out.
The liver completely abandons its specialized identity when it gets sick.
The peaks just flatten out almost entirely. The unique low sites, which are heavily enriched for critical liver functions like lipid metabolism and detoxification, they get inappropriately redacted and shut down.
And the unique high sites?
They are supposed to silence genes the liver shouldn't use, but they lose their redaction and mistakenly open up.
Let me push back on this a bit, though. If you look at someone with non-alcoholic fatty liver disease, they usually have excess fat in the liver, there's inflammation, there are physical changes.
Right.
Is this epigenetic amnesia just a symptom? Like, is the DNA simply reacting to the fact that it's sitting in a fatty, inflamed environment? Or is the software corruption directly tied to how sick the organ actually is?
Well, the researchers analyzed a cohort of 341 patients with NAFLD to answer exactly that.
Okay, what did they find?
They calculated an epigenetic information loss score for each patient and mapped it against their clinical traits. The loss of epigenetic information correlated very strongly with the fibrosis grade.
Fibrosis being the actual physical scarring of the liver tissue.
Exactly. The structural, irreversible damage to the organ. The epigenetic amnesia tracked tightly with the fibrosis grade, much more strongly than it correlated with the patient's age, their BMI, or even whether they had type 2
So
diabetes.
the sicker the liver was structurally, the more severe the cellular amnesia.
Yes.
That sets up a classic chicken or egg scenario. Is the liver getting scarred, which causes the DNA to get blurry and confused? Or is the DNA software getting corrupted first? And that corruption is what causes the liver to fail and scar.
And this is where the researchers utilize a really clever methodology called Mendelian randomization.
I love Mendelian randomization. It's essentially nature's own randomized clinical trial.
Exactly.
Right, because you can't, like, purposefully corrupt someone's liver DNA in a lab to see if it causes disease. That would be completely unethical. So how does nature's trial work in this context?
Well, Mendelian randomization looks at genetic variants that you were born with, variants that are randomly assigned at conception.
Okay.
If a specific genetic variant dictates that you will naturally have lower methylation at a certain site, and people with that variant also consistently develop liver disease earlier or age faster, it implies a causal direction.
Because the genetic trait was there from birth.
Right. So the epigenetic change must be driving the disease, not the other way around.
So if they cross-reference those data sets, I'm guessing the sites that lose their identity in a sick liver matched up with the sites that actually drive the aging process.
They found a massive overlap. Specifically, the unique high sites in the liver, the ones that are supposed to be locked down but mistakenly open up, they were significantly enriched for these known causal damaging methylation sites.
So it's not just the check engine light turning on when the liver gets sick.
No, not at all.
The epigenetic corruption is the engine actually seizing up. The loss of this specific liver identity is driving the biological aging of the organ.
Epigenetic information loss is an active mediator in the deterioration of the tissue.
Okay, we're at a pretty intense point in this story. Our cells are forgetting who they are, the blueprint is blurring, and that blur is actively causing the tissue to fail.
It sounds bleak.
It does. But we promised a reset button at the top of the deep dive. Let's get into the intervention. Can we actually stop this?
Yes. The researchers turned to mouse models to test a very specific metabolic intervention, intermittent fasting.
Oh, wow. Okay.
They used an alternate day fasting protocol, or ADF, for 30 days.
30 days of eating one day, fasting the next. What was the metric for success here? How do you even measure if the amnesia is reversing?
They measured DNA accessibility in the liver. Remember, methylation is a redaction marker.
Right, the sticky notes.
When a site is heavily methylated, the DNA is physically closed off. The cellular machinery just can't reach it.
And when it's unmethylated?
The DNA is open and accessible to be read. So measuring DNA accessibility gives us a really direct functional readout of the epigenetic state.
So they put the mice through 30 days of fasting and then looked at those specific liver-unique sites that had blurred out. What happened?
Well, let's look at the unique low sites first.
The ones that should be open.
Right. These are the sites that should be wide open in a healthy liver, but during disease, they regress to the mean and get inappropriately shut down. Following the alternate day fasting, the researchers found a 12-fold enrichment in increased accessibility at these unique low sites.
A 12-fold enrichment. Instead of just saying 12-fold, imagine a massive library that had its doors chained shut for years because the blueprints were deemed unnecessary.
That's a great image.
And suddenly you have 12 times as many workers aggressively prying those doors off the hinges to get back to the original instructions.
They opened back up. It's dramatic. And the reverse happened at the unique high sites.
The ones that should be locked down.
Exactly. The ones that mistakenly open up during disease. Following the fasting protocol, these unique high sites showed a massive enrichment in decreased accessibility. They closed back down.
The peaks and valleys came back.
Yes.
The fasting literally forced the epigenome to diverge back away from the blurry mean. It restored that sharp, specialized contrast.
It partially restored the youthful, specialized state of the liver's epigenetic software. And the functional relevance of this is just crucial.
How so?
When they analyzed exactly which sites were being pried back open, they found they were heavily enriched for binding sites of a specific protein called PPR-alpha.
PPR-alpha. How does that fit into the puzzle?
PPR-alpha is a master regulator of the fasting response and lipid metabolism in the liver.
Okay.
When the body enters a fasted state, PPR-alpha is the machinery that ramps up to burn fat for energy.
But how does a metabolic regulator actually fix the DNA software? Does it just, like, yell at the cell to do its job?
No, it's a very physical mechanism. Proteins like PPAR-alpha bind directly to the DNA strand at very specific regions.
Okay.
And when they bind, they act as beacons, recruiting other specialized enzymes whose entire job is to physically strip away those erroneous methyl groups, the redaction markers.
Wow.
The fasting creates the demand for PPAR-alpha, and PPAR-alpha physically clears the epigenetic clutter so the cell can function.
But wait, let me be the skeptic for a second. If I put a mouse or a person on alternate day fasting for a month, their whole system changes, right?
Of course.
Weight drops, hormones shift, immune cells move out of the liver. Are the liver cells really doing this software update themselves, or are they just passively reacting to the fact that there's less fat surrounding them?
That is the ultimate hurdle in this kind of research. It's the problem of bulk tissue analysis.
Because there's too much going on.
Exactly. To prove that this software reset is driven by the cells themselves, the researchers had to take the rest of the body completely out of the equation.
How do you do that?
They performed an ex vivo hepatocyte experiment.
Ex vivo, meaning out of the living organism. So they took liver cells hepatocytes out of a mouse and just put them in a petri dish.
Isolated in a dish, no weight loss possible, no immune system signaling, just the liver cell.
That's brilliant.
They then treated these isolated cells with 2 synthetic compounds designed specifically to mimic the fasting signal.
Like chemical fasting.
Exactly. One compound activated the stress receptor associated with fasting, and the other directly activated that PPAR-alpha pathway we just discussed. They chemically tricked the cells in the dish into thinking the body was starving.
And without the rest of the body there to help, could the isolated cells clear the clutter?
The results were definitive. Even completely isolated, these hepatocytes restored their unique epigenetic state.
Are you serious?
Yes. The genetic enhancers that were activated by these fasting mimic compounds mapped almost exclusively to the unique lowsites.
The ones that needed to be opened.
In fact, out of 143 activated enhancers they analyzed with one of the treatments, all 143 of them mapped to unique lowsites.
100% of the activity was dedicated to opening up the correct tissue-specific blueprint.
It proves that the cells themselves possess the intrinsic power and the active machinery to fix their own software.
That is mind-blowing.
This repair mechanism is cell autonomous. The memory of their youthful identity isn't permanently erased by aging or disease. It is dormant.
Dormant. Wow.
And a targeted metabolic trigger, even delivered chemically to isolated cells in a dish, is enough to initiate the physical restoration of that memory.
Let's bring this all together because the implications here are massive.
Yeah.
We started with the concept of cellular amnesia. Over time, as we age or face chronic stressors like sun damage or metabolic disease, the pristine, highly specialized software in our cells gets blurry.
The distinct instructions that make a liver cell regress to a generic pan-tissue average.
And through tools like Mendelian randomization, the data suggests this loss of epigenetic information isn't just a byproduct of a failing organ. It is a causal driver of the aging process itself.
The software corruption causes the biological decline.
But the incredible twist is that the corruption is reversible. By using a metabolic intervention like alternate day fasting, researchers hit the reset button.
They did.
They proved that our cells possess the internal machinery like PPAR-alpha to physically pry off those erroneous redaction markers, diverge away from that blurry average, and restore their sharp, specialized blueprint.
It fundamentally changes how we view lifestyle interventions.
Yeah.
We often think of diet or fasting purely in terms of mechanical outputs, calories in, calories out, burning fat, reducing load.
Right.
But this research shows that these physiological states are actually speaking a molecular language.
Think about your own body right now, to you listening. Your lifestyle choices, the periods where you might practice intermittent fasting, or how you manage your metabolic health. You aren't just changing the physical composition of your body.
No, you're doing much more than that.
You are communicating directly with the DNA inside your organs. You are sending a distinct metabolic signal that literally forces your cells to dig up their original blueprints, clear away the redaction markers, and remember how to be young and healthy.
The body's ability to maintain its identity is an active, ongoing process. The blueprint is always there, beneath the blur.
Which leaves us with a really fascinating thread to pull on. If our cells have this dormant memory of their youth and a memory of their optimal specialized state, and if that memory can be coaxed back to the surface with something as simple and ancient as repeated fasting.
It raises a profound question about our biological potential.
Exactly. What other dormant memories, protective mechanisms, or regenerative capabilities are hiding in the billions of letters of our DNA, just waiting for the exact right environmental trigger to wake them up?